It's my pleasure to have Dr. David here virtually for a presentation, 1 hour. For a quick introduction, Dr. David won Nobel Prize in 1975 for his work on discoveries concerning the interaction between human viruses and genetic material of the cell. I'm going to give the floor away for Professor David.
I'm very glad to have the opportunity to talk with you, what is for me in the morning and for you in the evening. The talk I'm going to give is mainly focused on early events in my scientific life, and it will be relatively short because I do want to leave opportunities for the audience to ask questions. But let me start by telling you a bit about myself today. I am an 86-year-old semi-retired professor who at age 80 decided to close my laboratory and live out my life as a consultant to nonprofit organizations and to companies. I have discovered that living through my 80s, I've gotten to know my doctors very well. Parts of me have acted up that I hardly even knew I owned. And I just finished a bout with COVID for the third time. Let me tell you that going to weddings is a good way to be exposed to the virus. But if I look back, the most remarkable thing in my life is that I received the Nobel Prize when I was 37 years old. So next year I will be celebrating 50 years as a laureate. Not many recipients get a chance to live with a prize for 50 years. And getting the prize at such a young age was testament to decisions that I made as a student. And I want to look back at those decisions because they were so seminal and yet so impulsive. The first was a decision teed up for me by my mother during my high school years. So let me tell you a word about her. She was a brilliant woman who dominated our family. And at the time, women were expected to be subservient to their husbands. Remember, I was born in 1938. And so successful professional women were rare. My mother went to a special New York City public high school and won a scholarship to New York University. She married when she graduated, but graduated into the Depression of the 1930s and was lucky to find a job in the garment business. My father had a successful small business of producing women's coats and suits that did well even during the Depression. He was an orphan. He never went to college. He only graduated from high school because his sisters dropped out of school and worked. They earned enough that their one brother could get a basic education. After my brother was born in 1942, my mother made the remarkable decision to return to school and earn a graduate degree in experimental psychology. She then taught at the New School for Social Research in New York City and at Sarah Lawrence College, where she was more or less ceremonially tenured at the age of 62. But back to the decision that I was faced with. This was when I was about 16 years of age, a junior in high school. So at the new school, one spring day, my mother saw a poster advertising a summer school for high school students at the Jackson Laboratory in Bar Harbor, Maine. She brought it home to me and suggested that I consider spending the following summer at the Jackson Lab. Jackson, as you may know, is a laboratory devoted to the study of mouse genetics. And having no better plan for the summer, I applied and was accepted, and the experience provided the direction for the whole rest of my life. At Jack's Lab, as it was known, I and 25 other students listened to lectures, swam in the very cold waters of the Atlantic Ocean, and most importantly, did some little research projects with 3 of the faculty who were resident at the lab for the summer. These projects were of minimal complexity, but gave us data, and by the end of the summer, we had discovered some new little things about mouse genetics. To me, that was remarkable. As a high school junior, knowing little about mouse genes, I was able to make discoveries. Research biology was available to me. It was my first encounter with data, and I loved it. I then and there made the decision that I would spend my life at this endeavor, a decision from which I never deviated. To this day, my greatest pleasure is to produce or be handed data about biological processes that was produced in a controlled experiment. There is a lesson here. In your teenage years, it's not too early to establish your trajectory in life. Look at all the athletes assembled in Paris right now. They generally started their careers even earlier. I have met many scientists who had a chance to do research before they went to college, and they all point to that as an experience that was seminal in their lives. Let me jump ahead to when I entered graduate school. That was in 1960, just 7 years after the discovery of the structure of DNA. Now, 7 years is a long time today. A discovery that would be old news 7 years after it was made. The discovery of the structure of DNA marked the advent of the era of molecular biology, but it was so revolutionary that in the 7 years that ensued, there was surprisingly little development of molecular biology. I was fully aware of that development, and I had gone to MIT as a graduate student, where Salvador Luria, one of the founders of molecular biology, was just starting one of the first molecular biology groups, teaching groups in America. My first job, of course, was to search for a mentor. And as I did that, I realized that the one organism that molecular biologists had focused on was bacterial viruses. They were simple, easily manipulated, and cheap to work with. However, they were the object of the past work, and I was thinking about my future. I loved working with mice, but there's not much you can do in molecular biology with mice in 1960. It occurred to me that animal viruses might be the right research object to start on, and I asked Luria if that made sense. He didn't know, but he suggested a plan for me to find out for myself. Luria arranged for me to spend the following summer working with an animal virologist and taking the animal virus course at the end of the summer at Cold Spring Harbor. I did that and was convinced that there was much to be done with animal viruses that might teach us about the unique properties of animal cells. To pursue my new direction, I moved to the Rockefeller University in New York City and began research with Dr. Richard Franklin, a young faculty member at Rockefeller. I chose to focus on RNA viruses, not because I realized that RNA biology was to become one of the most exciting developments in biology, but because Richard Franklin, my mentor, worked with them. Poliovirus became the object of my concern, and I quickly learned how to study its RNA genome and the proteins it encoded. I continued to focus on poliovirus and its relatives for the 8 ensuing years, doing my thesis quickly and a couple of postdoctoral stints, and then moving to the West Coast to the Salk Institute and ending as a faculty member at MIT in 1968. At MIT, I extended our work to cover other kinds of RNA viruses, discovering a negative strand RNA virus in the process. But there was one class of RNA viruses that remained enigmatic. They were the RNA viruses that caused cancer. Cancer itself was an enigmatic disease. We didn't know the first thing about it, and the first thing was to find out whether it was cell development gone wild or was it a genetic problem. I set out to understand the unique characteristics of RNA tumor viruses. My approach to a new class of viruses had become biochemical at that point. I found the poliovirus RNA polymerase and continued to study viruses by finding their polymerases and studying them. And we could discover in that way the unique properties of viruses. Some viruses had polymerases even in the virus particle, making them particularly accessible. In this work, I was following the lead that had been provided many years previously by a great cancer virologist, Howard Temin. Howard was convinced that RNA tumor viruses must have a phase in which their RNA genome was copied into DNA. The idea that RNA could be copied into DNA was chemically reasonable. They're very similar molecules, but it violated what had become the dogma of biology, that information flowed from DNA to RNA to protein, but not back in the other direction. Howard had tried for some 10 years to prove his idea, but he could not produce an experiment that convinced anyone else. The problem was really technology. The technology of molecular biology was so rudimentary that there was no experiment that could convince people. So because I knew Howard, I had actually met him at my summer in Jackson lab. I took seriously his heretical idea that RNA might be copied into DNA. This led me to do the experiment that won me the Nobel Prize. I looked for an enzyme that could copy RNA into DNA in the virus particles themselves. And sure enough, there it was. It became called the reverse transcriptase, and it was found in the virus particles. The actual experiment that demonstrated this, I did in just a couple of days because I had been doing that kind of biochemistry for years. Howard also found the enzyme, and we published back to back in Nature. That was in 1970. The Nobel Prize for me and for Howard came in 1975, so quickly that I was awakened from a deep sleep by a call from my wife, who was in Europe at the scientific meeting where she heard that I was to be given that award. Why was the Nobel Prize given so quickly for this discovery when it usually goes for work done many years previously? It was because of the implications of the discovery. The very simple experiment I had done opened vistas for research by many investigators. There was a strong implication that cancer was a genetic issue and that we needed to look for genes that were causing cancer. It galvanized research that ultimately led to the discovery of oncogenes. There was a strong impetus for the biotech industry because the enzyme could copy messenger RNAs into DNA and thus capture the genetic information for any gene. And as genomes were sequenced, it became clear that much of the higher cell genome arose by reverse transcription. There have also been many spin-offs, enzymes found that use reverse transcription as part of their mechanism. Now, there are many morals to this story, but the one I want to stress is that students need to take their education into their own hands. At every step in my career, I've had decisions to make that involve big steps into new directions. And by not being afraid to take those steps, by not looking back, but continually looking forward, I have kept the momentum of my evolving life. I was lucky that the world I entered was one of great possibilities and little understanding. There were very big questions open in 1960. What were the roles of RNA? What was the nature of cancer? Could we build an industry around the discoveries of molecular biology? In my lifetime, these questions have been answered. But there are still big questions to be investigated in the lifetimes of those who are young today. How does the brain work? What drives the aging process? What is the nature of dark energy and dark matter? After the Nobel Prize, my life has taken many new paths. I moved from being solely a virologist to also focusing on cancer research. I moved further to immunology, and the most exciting work for my laboratory over the middle years of my life was in immunology. While never leaving active research, I also became an academic administrator, starting the Whitehead Institute in association with MIT, and then heading the Rockefeller University and the California Institute of Technology. I would be glad to talk about any of this, but I would rather do it as answers to your questions than going on to be just a lecturer. So let me thank you for your attention and encourage you to ask questions. If you don't have questions, I have a few.
Thank you so much, Professor David, and allowing us to have time for questions. I'm very sure we have a lot of questions. I'm going to invite the students, invite the interested in the first row, and who would like to go first? Would you come forward?
Hello, thank you for your speech. It was so inspiring. Could you elaborate a bit on your work within the nonprofits and how you contribute to them? Because I'm really
I can't follow you. Speak just a little more slowly.
Hello, sorry. Could you elaborate a bit on your work with the nonprofits and how you contribute to them?
How I what them?
Contributed, like how you work with them.
How I
Your
work
work
with
and
them.
contribution with non-profits.
Yeah.
Yes. The nonprofits that I work with are mostly academic institutions, other universities, research institutes. And I've done two things. One is to help them develop their own particular style. I've been a director of the Broad Institute in Cambridge, Massachusetts for many, many years. Um, but the other is to encourage philanthropists to support nonprofit organizations, particularly in basic research. Philanthropists are playing an increasing role in the support of science in the United States and elsewhere, um, because we're getting this incredible crop of billionaires who have so much money that they can afford to support basic research uh, while they live a very active life. Um, and they need advice, they need direction, they need help because they find themselves supporting things that they don't understand as well as they do the industries that generated their wealth. And so I have been involved both in talking with and advising philanthropists and organizations like other universities and institutes. I have a special fondness for small institutions uh, because I went to a small college, Swarthmore College, and built a small research institute, the Whitehead Institute. And I believe that small institutions have tremendous freedom to take risks, to do unlikely things. And so I really encourage their development.
Thank you so much for your sharing. I was wondering, what do you think are your most precious life lessons from your many years of research and receiving the Nobel Prize?
Well, there are many lessons. Hold on just a second here. Yes, there are many lessons. But let me let me just take one. Uh, that we say that one of the rationales for basic research is that it generates practical knowledge, and that we don't know where that practical knowledge will come from. Hold on, my phone is acting up here. Hopefully it'll quit. And I, in fact, have not developed any drugs myself or practical applications of the research that we're doing. But on the other hand, other people have taken discoveries that we've made and then turned them into practical drugs. Let me give two examples. One is the inhibitors of reverse transcriptase. So we discovered the enzyme, and then the pharmaceutical industry discovered compounds that would inhibit the enzyme. And the place where that made the largest difference was in the AIDS epidemic, because AIDS turns out to be caused by HIV, which is a virus controlled by a reverse transcriptase. And so inhibitors of reverse transcriptase became inhibitors of HIV. And that has been a remarkable story of continual invention by the pharmaceutical industry, so that there are drugs today that I think are taken once every 6 months that can control HIV and allow people with AIDS to live a long and effective life. The second example is Gleevec. So in 1970, when I discovered the reverse transcriptase, I decided to use part of my laboratory to study viruses that cause cancer. And in particular, we found a virus. We didn't discover it, somebody else did, called the Abelson virus, which caused a very rapid, very flagrant mouse cancer. And so it was a good object for study because you could study it in over short periods of time. We found the oncogene in that virus and found that it carried out what was then a very unusual biochemical reaction, the phosphorylation of tyrosine. Tony Hunter found the same thing at the same time. And the remarkable thing was that the virus that we found had used an enzyme to phosphorylate tyrosine, which was the same enzyme that caused a human cancer. And again, the pharmaceutical industry started making inhibitors of that enzyme, and those inhibitors blocked a tumor, chronic myelogenous leukemia, in humans. It was actually the first and really the only magic drug that could completely block a human tumor and allow people to live, again, a normal, long life, even with the disease. So the reverse transcriptase inhibitors and Gleevec were spin-offs from research that we did because we were interested in the underlying biology, not because we thought we'd make drugs, but they did lead to drugs, and they led to very practical advances in the treatment of disease. And that is, in some ways, the most satisfying thing that has come out of our research, is that by following my own curiosity, I was able to make a significant difference in medical problems.
Hello, Dr. Baltimore. I am Pakin from Thailand.
Hello.
Do you have any advice for aspiring young researchers?
I guess the advice is to stick with it. Research is not an easy career. You have to convince people to give you the money to do work. You have to publish with some frequency. You have to learn how to run a laboratory. It's not an easy life, but it's an extremely satisfying one, because you get to wrestle with fascinating questions, and the results, as I said, can be very practical effects on disease and on society. Research is also an activity that requires a larger framework. You can't do it by yourself in a garage. You need a laboratory in an established institution like Cambridge University or like MIT. You need around you a tremendous amount of technology. And you need very bright young people to work with. And all of that means that the institutions of science are a very important part of the creation of science. And that has led me to become a university administrator, because I believe that universities are a very important part of the research enterprise. And I have, for 9 years, I was the president of Caltech. But all of that time at Caltech and at Rockefeller University and running the Whitehead Institute, I have maintained a research laboratory because it is research which I really care about. And it is possible to do many different things at the same time. There's a lot of time during a day, but you have to keep focused on what you care about and keep your priorities straight.
Thank you. Just one question from my end. If you can possibly tell us and compare your experiences with young researchers, senior researchers, other Nobel laureates, industry people, NGOs. Can you compare your experience with them?
I don't really understand what the question is.
Can you compare your experiences with different people in the range of young researchers to senior researchers, possibly other Nobel laureates and industry?
You know, as I have gotten to know various scientists, and I've met scientists all over the world, it's become clear to me that each one is a separate story. Scientists come from every part of society. Some come from wealthy backgrounds, some come from very poor backgrounds. Some had an easy job coming up through great universities. Others went to local schools and had to build their careers from very weak foundations. And that's Nobel laureates as well as the many other people in the scientific community. There is no established pathway. Each person has to find their own way. And each person has a different background to build on. I emphasized my mother's role in my life because that was seminal. It was extremely important that she had an appreciation for basic science, that she led me in academic directions. She understood me in that sense. But other people come from broken families where they don't have the opportunity to get parental involvement. And often it's a teacher who makes a huge difference, or somebody that they meet along the way. So I don't think there is any paradigm. There are just a whole lot of stories. And I'm always amazed when I meet take a Nobel laureate like Phil Sharp at MIT. Phil came from rural Kentucky. He grew up on a farm, a hardscrabble farm, as I understand, and had very little in the way of resources to build upon. But he found his way to a local college and then to the University of Illinois, which at the time was probably the best school in the world to study chemistry, which is what he did. And finally, he ended up at MIT. In fact, I hired him, and has had a remarkable career. Is in fact quite wealthy from biotech. So, so as I say, every story is different, and every individual is different, and we make up the whole scientific world.
Thank you so much. Do we have any questions left? We have one more question. Would you like to come forward, please?
Dr. Baltimore. Thank you so much for your speech. I was just wondering if you could elaborate some more on the significance of RNA research in understanding and treating cancer.
Yeah, RNA has been a sort of stepchild of biology. When Watson and Crick discovered the structure of DNA, people who knew about DNA and RNA wondered whether the structure of RNA was similar to the structure of DNA, a double helix. But you couldn't find any evidence that RNA formed a double helix. Actually, we discovered later that RNA does form a double helix, but the RNA of RNA viruses. But the main structure of RNA was as a single strand. And DNA had a single obvious role to play, that is, as the storage house of genetic information in biological systems. But RNA seemed to be everywhere. There was RNA in the cytoplasm, there was RNA in the nucleus, there were long RNAs, there were short RNAs. There was no consistency to RNA. And for the first, I would say 20 years after the discovery of the structure of DNA, RNA was a sort of stepchild. There were always RNA viruses, and in RNA viruses, RNA could act as genetic material as well as a template for the synthesis of proteins. And so we knew that RNA could serve complex functions, but we didn't know what all the RNA in the cell was doing. And there was a hint that was very hard to follow, and that was the ribosome. Ribosomes had huge amounts of RNA in them, large RNAs, clearly a structural component of the ribosome. But did it do more than that? We didn't know. And, from the first 10 years after the discovery of the structure of DNA, we didn't know about messenger RNA. A little less than 10 years, 8 years. Messenger RNA was discovered in 1961. And the important thing in the discovery of messenger RNA was to differentiate messenger RNA from ribosomal RNA, because people thought that maybe the ribosomal RNA encoded protein. But in fact, ribosomal RNA serves chemical functions as well as structural functions, but doesn't encode proteins. But in the last, 20 or 30 years, we've discovered more and more functions of RNA. First function of RNA was found was as the adapter between messenger RNA and protein, the transfer RNAs. But now we see microRNAs, we see long non-coding RNAs. There are all sorts of different functions of RNA, and we believe that there may have been an RNA world in which there was no DNA, and in which RNA served catalytic roles, served enzymatic roles, and served structural roles, as well as genetic roles, and that maybe RNA viruses are particularly a remnant of the RNA world. And so over the years since the discovery of the structure of DNA, RNA has become more and more and more important to the point where today RNA is part of almost every story that you hear. And new exciting roles of RNA are coming up continually. It's particularly true at Caltech, where we have people who focus on RNA.
Thank you so much for your answer. I think I might have fixed the mic. Can you hear us better?
Yes, I can.
Okay. I propose, can we take 5 minutes? We have few of our high school researchers and first year college students. Is it a good idea if they propose their title, the research title they worked on, so you can share your thoughts?
Sure.
Would anyone like to go first? I invite anyone working on RNA related, medicine related, to just share your title you presented, the research title you presented with CCIR, the the one you the one you worked on. So he gives you more info on that. I would say firstly someone related to medicine, physiology. Have anyone? Or any other questions? I think we have 10 minutes of time.
Well, there's one question that people often ask, and that no one has asked, and that is, what is the most negative event in my life? I've had many scientific discoveries and activities which are very positive, but has there been a negative event? And the answer is yes, there has been. There was a controversy that started over work of a collaborator of mine, whose name was Teresa Imanishikari. And Teresa was a very good immunologist. She knew how to do things that I simply did not know how to do. And so we worked together on a number of papers in immunology. But then a postdoc in her laboratory accused her of fabricating research. This was very early on, when that kind of fabrication was rare. And because she was actually a foreigner, she had come from Japan and from Europe, and was, I thought, being treated most unfairly. I defended her. This escalated into the Congress of the United States, where a congressman took on the charge against Teresa, and I had to defend her in the Congress of the United States. The problem lasted for 10 years until she and I were entirely exonerated as not having done or anything wrong, except being a little sloppy. And she was, I must say, a little sloppy. But she knew her science brilliantly, and was simply way ahead of people in thinking about what she was doing. And so for 10 years, this hung over my head. It caused me to lose administrative positions. It caused me to lose friends. The loss of friends was one of the worst parts of the experience. But finally, I came out at the end, able to continue my life. And I was lucky to have people who still believed in me.
Thank you for sharing the question. Thank you for sharing your experience. We have one more question from the audience. Can I redirect if you, or I'll try to redirect if you. Professor, we have a student who would like to share their current research title and get your insight on it.
Yes.
So right now, it's still ongoing, of course, but I am currently researching on the inhibition of actin rearrangement in brain-eating amoebae. To be more exact, the Palamutium andilaris species. Yeah, I am currently facing a problem with the part of I already have the parts of the data, but I still have to, of course, figure it out, like how to write it all out.
Well, actin is one of the most important proteins in biology. It plays the obvious roles of being partnered with myosin in making muscles work, but is much more important than that, particularly in cell movements. And so trying to understand actin anywhere is critical. I don't know what the organism that you were talking about is, but
It was an amoeba.
It's
It
an
was a
amoeba.
great amoeba,
Yeah.
and the purpose of the research was to see whether it could inhibit the movement of the amoeba and therefore limit whole cell death, as it would limit the phagocytosis process of the amoeba.
Well, yeah, so that's a very practical goal, but you're working with a very basic protein, not basic in the sense of pH, but basic in the sense of fundamental. And what it really shows is that today you can do research on any organism. When I started out, as I said, the major organism that we worked on, or that the molecular biologists worked on, was bacterial viruses. But you can't learn the whole world of biology through bacterial viruses. You've got to be able to study each individual species for its own characteristics. And we couldn't do that because we just didn't have the tools in 1960 to do that. As I said, I would have liked to have worked directly on mice, but I couldn't begin to work on mice until after 1975, when recombinant DNA methods were developed so that you could then work on higher cells. And what's happened in just the last 20 years is that, particularly with CRISPR-Cas9 and other technologies, you can now work on any organism at very fundamental levels. And that has opened up vistas into biology that we never had before. And so I would encourage all of you to think about what are the characteristics of particular organisms that make them so interesting. The colors of butterflies, for instance, or the many, many other things in biology. Because you can ask very fundamental questions about those now that you couldn't 20 years ago.
Thank you so much. We have one more question from the audience.
Yeah.
I think the mic is a little better. You can give it a shot. Yeah, that should be fine.
Hi, Dr. Baltimore, can you hear me?
Yes, I can. Thank you.
Hey, one of the tools which has opened up for biology is AI and computing processing. What are your thoughts on that tool in general, but also on brain research in particular?
Well, I sit there and marvel at what AI is able to do, but I must say that I really don't understand it. Although I remember when AI was started, it was when I was at MIT as a student, where the first AI people were working at MIT. And I followed it for years and years, and nothing happened. AI was a thing unto itself, and it had no important implications. And then suddenly it became the center of everybody's attention. It's been most remarkable. But I can't say that I have any particular insights into what AI can do. Brain research is another thing which I have followed from a distance for many, many years. And we're seeing a tremendous renaissance in neuroscience today. Questions that were unapproachable 10, 20 years ago are suddenly available. We're finding brain circuits down at the level of single cells. And single cells are where you have to investigate brain function, because the brain has so many functions that are all intertwined together in the brain. But there are still very, very fundamental questions about the brain. What is the unit of understanding? What is the unit of memory? What is the unit of consciousness? And so I see brain research as very exciting for the future, but also extremely difficult because it requires working at a very microscopic level on questions that have results at a very macroscopic level, like in behavior, animal behavior.
Thank you so much for your answer. We have one law student who would like to share a slightly different research topic and get your insights, please.
Hello, Dr. David. So my research was focused on the interplay of hippocampus and basal ganglia, and how the interplay of these two regions of the brain facilitate the formation of habit and goal-oriented behavior, which is assisted by the mesolimbic dopamine pathway. So I just wanted to get your insights a little bit on the further direction that I could go with this paper, because a lot of this was based on expansion of literature, because I couldn't do a lot of quantitative data on it. So if I wanted to enhance my paper a bit further, which direction would you suggest for me to go into?
This is really a good question where students face lack of data and they do quantitative literature review. Do you have any recommendations for them to go ahead?
Yeah, I'm afraid that in that kind of research, I have no particular experience or understanding. But as I said, I think that kind of research in particular requires going down to the single cell level. Single cell RNA-seq methodologies are revolutionizing our understanding of the deep complexity of the brain. So I would suggest that you develop those skills. But beyond that, I don't know much.
Thank you so much. Do you have any final words for our aspiring students before we close up?
Stick with it. The most exciting career you can possibly have is a career in research, is looking into the unknown and continually trying to make sense of it. But as I said, it's not an easy life, but it's a very satisfying one.
Thank you so much. We really appreciate your time. Have a nice day. Thank you.
And thank you all for your attention.